Hello, everyone, and welcome to our event, The Science of Addiction. I'm Carmel Shachar, the Executive Director of the Petrie-Flom Center for Health Law Policy, Biotechnology, and Bioethics at Harvard Law School. It is my pleasure to welcome you to today's event. This event is part of the project on Law and Applied Neuroscience, which is a collaboration between the Center for Law, Brain and Behavior at Massachusetts General Hospital, as well as the Petrie-Flom Center. We're very honored and appreciative of this long-standing collaboration with our colleagues across the river. Before we get into the substance of today's event, a few logistical items. We strongly encourage you to submit questions for our speaker today. Please use the Zoom Q&A feature, which is found in the meeting controls at the bottom of your screen to submit questions. That's by far and away the best way to get your questions to our moderator and to our speaker. If you would like, you could also join the conversation or even submit questions on Twitter. We're at Petrie-Flom, and you can use the hashtag LawNeuro. Again, that's hashtag LawNeuro. If you do that, we'll see it and we'll pull the question and put it into the Zoom Q&A feature. On the other hand, if you have any technical issues or problems, please email us at petrie-flom@law.harvard.edu. Again, that's petrie-flom@law.harvard.edu, and we'll do our best to fix your problem. We will be sharing the fully captioned event video with all registrants within 1 to 2 weeks. So if you like this event and you want to relive it, I completely understand. I'll probably feel the same way. You can watch the video then. If you have friends who are interested but couldn't make it today, please do share the video with them when it's publicly posted. If you like what we do in this event, and I hope you do, and you're interested in our further activities, please sign up for our newsletter. It comes out only twice a month, so it does not clog your inbox. It's the best way to find out about scholarship published by center affiliates, what's going on our blog, upcoming events. Speaking of our blog, if you're interested in further in-depth health law and policy and bioethics conversations, please feel free to check out our blog, The Bill of Health. We just finished up a wonderful digital symposium on the framing of adoption as an alternative to abortion, unpacking that and exploring that. If you like our events, please check out some of our upcoming events. We have a great one on Friday looking at the impact on vulnerable individuals of this new phase of the pandemic, of what's being referred to as the new normal. I want to also do a big plug for the Center for Law, Brain and Behavior. We are busy, but they are somehow even busier. They have fantastic work. If you're coming from the PFC side, please check them out at CLBB.org. Find them on Twitter at MGHCLBB or on Facebook, also facebook.com/MGHCLBB. With that, I want to introduce our guide for today, who is Dr. Stephanie Tabashneck. She is a psychologist and attorney. There can be no better figure standing at the intersection of law and neuroscience, which is in part why she is a senior fellow in Law and Applied Neuroscience, which is a joint position between the Center for Law, Brain and Behavior and the Petrie-Flom Center. Dr. Tabashneck, I'd love to hand the event over to you.
Thank you so much, Carmel. I really appreciate it. I am so excited to be here. I think this topic is really important because when we understand the science of addiction, we can set appropriate expectations, we can move away from harsh shaming responses that are unproductive and don't work, and encourage smart policies within public health, criminal law, juvenile justice, parole, and family law. Today I'm here joined with Dr. Eden Evins. I am elated that she is here. If I were to read all of her accomplishments, we would be here the full hour. So what I will tell you is that Dr. Evins is an expert in the area of addiction. She's a psychiatrist and runs the Center for Addiction Medicine at MGH. She's also, today she's going to be speaking about the science of addiction, and she's going to be covering a multitude of topics, including neuroscience, prognosis, and treatment. After she presents, we're going to have a rich discussion about the implications for substance use disorders and public policy and public health and other domains. And please make sure that you put your questions in the Q&A, and we're going to try to get to as many as we can. So with that, I'm going to hand the presentation over to Dr. Evins. Thank you.
Thank you so much, Stephanie, and thank you, Carmel, and thank you everybody for being here. It's just such a pleasure to speak with folks who are in the intersection of policy and science and law, where I feel like some of the work that's done on the science side actually can benefit people and society. So I'm going to share my screen and get started. So unusually, I've called this good news on addiction treatment, because really addiction is a good prognosis disorder, much to the contrary to what is portrayed in the media. It is a major addiction crisis, but with treatment, it is a good prognosis disorder, and I'll talk a bit about why. But first, I want to talk about why people get involved with drugs. So why do people take drugs in the first place? Well, they like what it does to their brains. And how does that work? Well, there are many neurotransmitters involved, but dopamine is pretty central. It's not only dopamine, but this is a cartoon of a synapse, to basically letting you know this is where we start. These are two nerve cells in the brain. Dopamine gets released and binds the postsynaptic receptor, and this is what gives drugs that gives you the pleasurable sensation. And why do we have this? Well, dopamine is released in response to things that are good for us. So food and sex, things that are good for us as people and as a species. These are sort of experimental single neuron recordings of dopamine spiking when people are when mice are exposed to food or sex. You see the female is present and the dopamine rises. What we have, though, is drugs of abuse that bind and cause to these dopamine transporters and cause a huge amount of release of dopamine, such that amphetamine causes 10-fold higher dopamine release, cocaine 4-fold higher dopamine release than food or sex, nicotine 2.25-fold higher, morphine almost twice as much. So drugs, basically drugs of abuse, and to some extent, somebody already asked a question about sugar, to some extent sort of palatable, high nutrient density foods also cause this dopamine spike. THC also, because people talk about THC not being addictive, I thought I'd put that in here as it brings it up almost 50% higher than food or sex. So THC is the addictive component in cannabis. Excuse me. And why do some people, I'm going to keep coming back to it, why do some people get in trouble with drugs of abuse and others don't? That's the question I'm asked like at Thanksgiving dinner every year. And one thing that we know is people are born with inherent differences in their pleasurable responses to drugs of abuse, to how much they like drugs of abuse. So people can have high density of dopamine receptors and low density of dopamine receptors. Basically, just you take a large number of people, there's going to be a variation, and those in the dopamine receptor level really mediated how well they liked amphetamine. Those with higher dopamine receptor levels really didn't like it, and those with lower dopamine receptors liked it more on average, right? So this is something you come into the world with. Say those of us who have a high dopamine receptor level may be somewhat protected against addiction because we don't really find the first or second dose of a drug of abuse particularly pleasurable or less pleasurable, right? So just wanted to speak a bit more about genetics. This is one slide summarizing, a mountain of research, which is not been very informative until fairly recently. Really, in the past 15 years, we've found essentially common genetic risks, genetic risks that predispose people to actually initiating drugs of abuse. So you know, whether or not you'll even try a drug of abuse is to some extent impacted by genetics. And then there's a high genetic correlation between cannabis and tobacco initiation, right? So we probably aren't genetically programmed for a specific drug of abuse, although people may have a preferred drug of abuse, right? So not only initiation, but then other things that lead to addiction, such as the quantity and frequency of use, and then with ability to quit, so success at quitting. All of these have, you know, genetic correlations. And because psychiatric illnesses, we're not going to find a sort of a smoking gun, a single genetic hit that strongly predisposes folks, we don't think, to any sort of behavioral disorder, psychiatric or substance use. It looks like there are many, many genes that are involved, and their combination can significantly increase your risk for having a disorder. And those are called polygenic risk scores, right? So it's essentially many small effects that, when they are combined, significantly increase risk. And the same PRS, they're called, is associated with alcohol and cannabis use. We just had a paper showing the same PRS is associated with smoked tobacco use and vaped tobacco use. So not hugely surprising. So, you know, genes in a specific way have been found to contribute to vulnerability for addictive behavior. We've known for a long time that addiction is highly heritable, but it's a little bit hard, has been hard to disentangle that part of your inheritance that mediates your environment versus that mediates your behavior specifically. And so this latter piece is starting to be fleshed out. So we know that when we meet, say, in a courtroom or somewhere, someone who has an addiction, they came into the world quite likely with a vulnerability, a genetic vulnerability, in addition to whatever environmental risks they may have had. This is just showing another set of risks for addiction. People with psychiatric comorbidities have extremely high risk for addiction. You know, I often talk to the kids with bipolar disorder who are diagnosed by my colleagues, and, you know, tell teenagers who've been diagnosed with bipolar disorder that they just can't sort of experiment with alcohol and drugs in high school like their friends are doing, and it's not fair and it stinks, but they've got a 50-50 chance of becoming addicted should they start using that early in their life. And so here you see people who've had mania, you know, have a 35% risk of an illicit drug use disorder. That's not even just, you know, alcohol. And here you see schizophrenia and depression with an 80% and a 50% likelihood of nicotine addiction compared to a 24% risk in the general population. So these are substantial additive risks over and above what is experienced with those in the general population. So it's just another risk factor that people, it's not choice, right? It's something that they come into the world with, you know, and even at a more basic level, we can look at factors like impulsivity. And Walter Mischel, from 1968 to 1974, performed an experiment amongst 4-year-olds who lived in and around Palo Alto. He was at Stanford. And wanted to test inhibitory control in kids, and then follow kids longitudinally and look at the impact on outcomes later in life. And so I just want to show you a couple of minutes of the marshmallow test. And so the setup is he brought kids in basically starting from his kids' preschool, and they sat them down. It's very simple. They put a marshmallow on the table, and then if the kids who didn't like marshmallows, there was a piece of chocolate, and said, you can eat it now. And I'm going to go check something in my car, is usually what he said, and I'll be back. And if you don't eat it, you can have 2 when I come back. And he did this in about 540 kids. And interestingly, everyone said they would wait until he came back. So let me switch this. So how do I do this? I'm going to stop share, and then I'm going to reshare with this other screen and show you the marshmallow test.
Okay, sit in that chair. All right, here's the deal. Marshmallow for you. You can either wait, and I'll give you another one if you wait, or you can eat it now. When I come back, I'll give you another one, so then you'll have two. But stay in here and stay in the chair till I come back, okay? All right. I'm gonna go do something and then I'll come back.
It smells yummy. It smells really good.
So it's up to you. You can have it now or you can wait. Okay? I'll be back. Stay in the chair, okay? Okay. So I'm gonna leave and then I'll come back, okay? So you can either eat it right now or you can wait. Either way, okay? Okay. How'd you do? Did you do good? You did? You wanted to eat it, didn't you? Yeah. So did I tell you to give you another one? Okay, now you can have both. You need them.
So while everyone said that they would wait for the second marshmallow, only 1/3 did. And then remarkably, over the next first 15 years and then at a 40-year follow-up, those who were able to delay gratification, right, at 4, wait 15 minutes for a second marshmallow, had higher SAT scores, over time made more money, had fewer divorces, had less addiction, less obesity, and then so those were the outcomes we knew until just a couple of years ago, when B.J. Casey did a remarkable study in which she brought people into the scanner. She picked people who those who did and didn't sort of weren't able to delay gratification, and they had differences in their prefrontal cortex. So they had greater ventral striatum activation when asked to suppress a response to a desired cue, and greater activation in the sort of just say no part of the brain, if you will, in the decision-making part of the brain or the prefrontal cortex. So they had greater executive control in the ones who were able to delay. And this is likely, these are the brain mechanisms that are likely to undergird essentially the ability to delay gratification on most things, but you know, and that's needed in everyday life, and is needed, you know, is likely highly protective against developing addiction. So we love to talk more about genetics in the discussion, but I want to talk about environmental impacts. So people who have addiction have a much more greater likelihood of coming into the world with these risk factors, but exposure to drugs also changes the brain. And I want to show you some examples of that. And so from the animal literature, right, just THC exposure basically changes essentially the density of neurons in the nucleus accumbens. Okay, so the animals that were exposed to just saline, salt water, can you see the difference in how dense the fibers are in the basically vehicle versus the low dose versus the high dose THC exposure, right? They're developing greater sort of neuronal density, and that's what these branches, so this mean branch number and branch length are greater in those exposed to THC, low and high dose, in this reward sensitive part of the brain. And the thinking is if this happens in adolescence, there's even a greater impact. And I think I have a picture of that. Now, colleagues here, Jody Gilman in my group, I decided to do this in people. So she enrolled kids in high school who did not have cannabis use disorder, but reported some use of cannabis, which is more and more common, obviously. And she found that marijuana users had greater density than controls. And that's what these little blobs mean, is this where there's a subtraction, a significant difference in the subtraction. So they had greater sort of nucleus accumbens and striatal neuronal density that correlated with how frequently they used per day. And you can see most people didn't smoke every day or use cannabis every day. But those who 1, 2, and 3 times a day, and how much they used per occasion. As you see, most people, you know, use less than 1 joint per occasion. This is mostly before recreational cannabis and all the edibles were available. So this really is an example in humans of replicating these animal data. The animal data, of course, it's not choice. These are randomly assigned to be exposed to marijuana. And these kids, you know, there was some choice involved, so there may be some other things, other factors than just exposure to THC. In fact, there surely will be, although we tried to control for all that we could. But so here, THC seems to change the brain. And this is the same study showing that basically even the shape of the nucleus accumbens or the amygdala is different in those who had been exposed to marijuana, but that were not addicted, right? So there's essentially a bit of a loss of what should be normal pruning. So it's bigger than usual. And really in adolescence, at these kids' age, pruning is their job. They want to prune their brains. And it seems to be that there was less pruning going on in the amygdala in the kids that correlated with how much they were using of cannabis. This is nicotine, so this isn't just a cannabis talk. So this is back to an animal study. This is a fantastic study showing that if you gave nicotine, small amounts of nicotine 2 weeks before you exposed animals to cocaine, that cocaine was more addictive at 4 levels at the area of gene unwrapping. This is histone deacetylase inhibition. So those who had nicotine started unwrapping their DNA in important areas faster when they were exposed to cocaine. They had greater immediate early gene expression. That's the FOSB. they had greater essentially learning going on, and then they became they preferred the part of the cage where the cocaine was given to them more quickly. So, you know, most people think cocaine doesn't need a lot of help being addictive, but here, just being exposed to some nicotine every day for 2 weeks, randomly assigned, increased essentially the rate of addiction to cocaine on all these markers of addiction. And interestingly, if you swap the order, if you gave cocaine first and then looked at the addictiveness of nicotine, it did not. It didn't work in the reverse order. There was no impact. On the other hand, this is essentially the same study design, THC pre-exposure, so the addictive component in marijuana or cannabis, increased nicotine self-administration, right? So the self-administration rose to a much greater level in those who had THC pre-treatment. This is again an animal study. THC pre-exposure increased heroin self-administration. So this is people going out and looking for essentially a very clean version of, you know, the gateway hypothesis, which has been largely, you know, it's been a difficult thing to prove in epidemiologic studies. So this is, we call this essentially neurobiologic priming because the gateway hypothesis has gotten such bad press, but it's essentially that pre-exposure increases the use of and likelihood of becoming addicted to other drugs. And so far we've seen it for THC and for nicotine. And here exposure in adults even impacts drug use in offspring. We've even got people in Mass General who are looking at, have found that pre-exposure impacts offspring of the opposite gender in the grandparent generation. And remarkably, I haven't labeled this terribly well, but the cross-generational effects of THC exposure only impacts if the exposure is in adolescence. So this adolescent period is very important. And so kids who've been exposed in adolescence are more vulnerable. And this is just to remind me to talk about sort of the lasting impact of exposure, because essentially with this dopamine firing, that essentially cements memories in place. Dopamine burst firing is a way for our brains to encode important, often evolutionarily important emotional memories. And here's basically a picture of the same person watching a nature video and a cocaine video. And this is a video, Anna Rose Childress, fantastic scientist, works in Philadelphia, and these videos she made were really of sort of geographically specific. This was back in the crack epidemic in Philly, so she took some videos in some relevant places. So you can see the amygdala lighting up when they're watching this cocaine video, which is not happening. So you see the rest of the pattern is similar of brain activation, but this amygdala activation is bright as can be to the cocaine video, and she argued that this was essentially specific to cocaine. Here we have, I'm going to have now a series of essentially brain pictures looking at different elements in the brain in those who have had use of drugs versus non. So in the top, you've got a normal control, and this is essentially a PET scan of dopamine transporters, which I showed you in a cartoon, the first slide, right? This is nice and healthy, red means more. So there's lots of nice dopamine transporters in this healthy brain. And here's someone who's used lots of methadone and just very different, right? So this is very likely an impact of drugs on the brain. And this may be some predisposed factors, right? So remember I showed you those with lower dopamine transporter levels liked methamphetamine more in one of the first slides I showed you. It's very likely not to be this level of degradation, this level of difference. This is likely a combination of a predisposing factor and an impact of drugs on the brain. So two hits, right? And these correlate with both motor performance and cognitive performance. So this loss of dopamine transporters in the methamphetamine users is very likely to result in memory impairment. And here, so these are metabolic scans now. So how well how much energy is being used in these brains? And this is basically a proxy for neuronal activity or function. And so in the top row of 4 are the controls. And just basically all you're asking you to do is sort of match the colors. And some of these arrows sort of show you a hint of really where to look. Okay, and this is prefrontal cortex. So in the other pictures I was showing you sort of reward regions of the brain. These are the sort of the inhibitory control regions of the brain, the brains that help, the parts of the brain that help those 4-year-olds wait for that second marshmallow and would help people to essentially delay gratification or think about their long-term goals and be able to use refusal skills if they're in a social situation or an environmental situation in which they need to say no to an offer of drugs or an ability to use drugs. All right. So major difference. And this is very old-fashioned language. I need to change that. So this is somebody with a cocaine use disorder. And then here you can see it's not specific to cocaine or methamphetamine. Here on the left, there are comparison or control subjects, and on the right are people who have either a cocaine use disorder. You can see, again, this is the deep structures of the brain. This is a methamphetamine use disorder, and here alcohol use disorder, which really works through a different mechanism, doesn't work directly on dopamine transporters. It really works through GABA, but does increase dopamine. You see an abnormality, which is quite likely to be a 2-hit phenomenon, both a predisposed factor, which this is not testing, and the result of exposure of the drug to the brain. Now, here's the good news slide, right? And we need much more data here. This is getting to be a little bit old, but so here's the control. And then here's someone with just 10 days of abstinence in a treatment program. Again, cocaine use disorder. But here you see some recovery at 100 days. Certainly does not have the activity of the control subject, but looks a lot better at 100 days. Now that's 3 months, right? And that really leads me into wanting to talk about treatment, abstinence, and you know, our society, and we have a whole treatment program built out for, you know, residential treatment and acute treatment, and we expect people, once they've quote unquote dried out, to be back to normal. And that's just not consistent. Even though we have a whole sort of treatment industry built around that, it's really not consistent with the science. You know, what we really advocate for is extensive treatment rather than intensive treatment. I mean, some people may need both, but we really need long-term treatment in order to build new habits and to let the brain recover. So I just want to review a little bit. You know, why can't people with addictions, you know, just say no or just quit? And it's really a 2-hit phenomenon, right? So people, the brain of someone who's not addicted has lots of capacity in the prefrontal cortical regions, the inhibitory control regions, the just say no regions of the brain. And the salience of drugs, you know, drugs of abuse, they're fun, and they don't have no saliency, but it's not, it tends to not outweigh inhibitory control, which brings in, you know, people's memories, their long-term goals, you know, what they have to lose should they go too far. And the addicted brain, we know, looks a bit like this, where the control regions are not working as well. That's what B.J. Casey showed when she compared the kids who were and were not able to wait that 15 minutes for the second marshmallow, right? Very big differences in the ability of the inhibitory control regions or the prefrontal cortical regions to kick in, and much more activation in the limbic regions, or which really code for the saliency, which is driven a bit by the memory of drug use, drives this activation in the saliency regions. and they end up using as opposed to that's go versus no go, right? So it's again, this two hits. They've got people with addiction, which is likely people who have had both a predisposition and the environmental exposure, have increased drive and reduced inhibitory control. And we think together that's what increases risk for addiction. Now, it's not to say that this is destiny. We certainly have treatments that work. Contingency management works. Maintenance treatment, often with agonist therapies, so therapies that work like the drug of abuse, but that stay in the brain in the body for longer. So, for example, for prescription opioid addiction, we've got excellent outcomes for half of people when they're prescribed Suboxone, and that may not sound great, but a 50% response rate in medicine is fantastic. You know, you don't get that for, you know, most illnesses, hypertension, gout. You know, pick an illness, a 50% success rate, a cure rate essentially, or long-term positive outcome rate is fantastic. And when you stop the treatment, the success rate drops considerably. And so this is an argument for maintenance treatment or long-term treatment. But a major problem is that most people with addictions don't get treatment. And that's where, you know, drug courts can just have a fantastic benefit both to the person and to society, right? And but, you know, as a physician, I'm very interested in both, but I'm very interested in the individual outcome, right? And you know, Suboxone's not the only thing. There's methadone, there's naltrexone. We have choices, all work for opiate use disorder. You know, I did a 1-year trial. For most people with addiction, you know, a 12-week treatment program is a long program. We randomized people who had been successful at a 12-week program for nicotine dependence with serious psychiatric illness to either stay on treatment for a year, or the medicine treatment, or stop the medication treatment at 12 months, which was standard, 12 weeks, which is standard, but continue a group therapy. And at the end of 1 year, those who had stayed on the partial agonist therapy had a 3-fold higher abstinence rate than those who had who've just stayed on behavioral treatment alone, right? So there was a 60% abstinence rate in those who stayed on meds plus behavioral therapy versus a 19% abstinence rate at 1 year. And this is for people with schizophrenia who are addicted to nicotine. So, you know, there's this tendency to you know, it's outdated, but to think that, well, you shouldn't use medicine for an addiction. Don't trade a drug for a drug. Well, it's just if you're a pragmatist, that's absolutely not the right thinking, because people can go back, they become not addicted anymore. They're taking an agonist in order to function and function well in society. And this is just a reminder that essentially drugs of abuse tend to bind to areas in the brain instead of an endogenous neurotransmitter, right? But they're just stronger. So THC binds to the CB1 receptor where anandamide, a neurotransmitter that's active largely in neuronal brain maturation. But just binds more tightly and binds longer. So it removes the specificity of anandamide. So often what we're using for treatment is say a partial agonist or a drug that binds tighter than the drug of abuse and stays around for longer. So it stops the cycle of dosing for an acute high. Just to remind you, most people who use drugs of abuse do not become addicted, right? The highest are heroin and nicotine, right? So tobacco and heroin or opioids, almost 33% of people who use tobacco become addicted. 22% of people who use heroin become addicted. Cannabis, this is it depends on, this is just an illustration that it depends on age of onset of use. If you start as an adult, you've got about a 9% chance of becoming addicted to marijuana. If you only use occasionally as an adolescent, you've got a 16% chance. If you use daily for a period of 2 weeks or longer in adolescence, you have anywhere from a 25 to a 40% chance of becoming addicted. So age matters, developmental stage matters. But just to look at some of these numbers, right? So cocaine is fewer than 20% become addicted. Alcohol, I think it's 15 to 16%. So it's important to keep in mind that use, even problem use, doesn't necessarily translate into a use disorder. So I just want to sort of stop here with this cartoon to give us time to chat, just to sort of underscore there's biology that you come into the world with as a risk. There's environment, which includes your upbringing, it includes trauma, and it includes exposure to drugs that both impact drug use, brain mechanisms, and addiction. And each of these arrows is meant to be a place where potentially we could intervene either with treatment or policy changes. And I'll end with brain pictures. So addiction, like other diseases, is heritable. It definitely changes biology. It is both preventable and treatable. Genes are not destiny, but if untreated, can last a lifetime. And so these are PET scans of a healthy heart and a diseased heart. This is sort of energy metabolism, and someone with and without cocaine use disorder. So I'll leave this one up, I guess, for a bit. Or Stephanie, should I just take the slides down at this point?
Yeah, if you wanna take them down. I'm seeing a number of questions in the Q&A, and I also have a number of questions. You know, one thing I was thinking about, and there was one question about this, is when we're talking about substance use disorder and the impact of genetics and environmental factors, how do we sort of tease those out, or can we tease those out?
We know way more than we knew before, and we don't have the puzzle figured out. We know that there are large risks and large effects, right? I don't know how to, you know, and we know that if someone's got a family history of addiction, then, or we've tested and their polygenic risk score indicates they're at risk for addiction, then those are excellent candidates for preventive treatment and for delay of onset of use. Just like anybody who shows a sign of liking drugs early, we've just made essentially a school intervention, a brief intervention for kids who have infractions, to try to shift the culture from exclusionary practices, they call them, but basically getting kids suspended. We want them to get them into a treatment program that's brief and in school so they can stay in school, because that's another sort of way to prevent kids from progressing.
Mm-hmm. That's great. And I would imagine with animal models, you can sort of isolate things like trauma and looking at that and teasing that out from genetics a bit. So in terms of this is a big question, a broad question, in terms of just broader public policy considerations, what do you think our policy gets right, and what do you think gets wrong? It gets wrong in terms of—
Stephanie, I think we need to have a conversation about that. Policy is above my pay grade in many ways, so I conjecture, you know, that. You know, I think policy-wise, you know, it's not a great idea. I mean, the drugs that are not the ones that. The drugs that are legal are the ones that have the biggest public health implications, right? Illegal drugs have the biggest legal implications, right? So we focus most on tobacco, alcohol, and now cannabis, because those are the ones with the highest prevalence, right? But what I think the legal system is much more involved in illicit drugs, both for their use, because their use is illicit, and that their change in behavior essentially is associated with other criminal behavior, right, and behavior disturbance. Yeah, I think that the degree to which we romanticize drugs, you know, and commercialize drugs of abuse, advertise them, is a major problem. Focus on punishment and not treatment is a major problem, and we don't incentivize prevention. Right, prevention efforts are not reimbursed through the healthcare system. They're just not prevalent in the schools. I mean, the school's approach is punishment, and that's assuming that we all start from the same level of sort of free will, if you will. and really, help and education and environmental modification early, I think will be hugely helpful in terms of prevention. And we're poised now to test some of that.
There's been, some countries that have sort of shifted their country's policy and how they approach substance use disorders and both, legal and illicit drug use. And those countries have seen decreases in overdose rates and decreases in substance use disorder rates because it sort of shifted that money towards treatment instead of incarceration. Do you think the science supports that sort of position, or what are your thoughts?
Again, that's above my pay grade, and I would put prevention and treatment together. I'm a treatment. My work is treatment, but prevention is an extremely powerful tool that we have not availed ourselves of adequately in this country.
And if there were some prevention programs that were implemented, what are your favorite prevention strategies or programs that you think would benefit people who are at risk of developing a substance use disorder?
Right. So first, I'm in favor of basically, having people make informed decisions. People don't know that they're at high risk, generally speaking. So people who've got family history, really, they need to understand that, so that as they're going through adolescence, they're making as informed choices as they can. So kids who get diagnosed with ADHD, with bipolar disorder, are extraordinarily high risk of becoming addicted. So if they do the usual experimentation that their friends are doing, they've got a higher risk of getting in trouble. So education, and then early intervention. So secondary prevention would mean if you get caught using a bit, bring people in, there's some nice tools. There's wonderful tools. The NIDA website has a nice tool for adolescents. I can put a link for that. Actually, the daughter of a friend of mine at NIDA made the video.
That's great.
It's a neuroscience video, and so it's absolutely, the science is correct. And so it puts it out there for kids in middle and high schools, so that they can make better choices.
I always find children and adolescents sort of complicated in this analysis, because I think, in my experience as a clinician and an evaluator, it's really hard to sort of, encourage kids to not use substances and not be effective. I mean, I just think of, I don't know if when you were a child, people saying, oh, drugs are bad, and now I'm, drugs are bad, and I just wonder, I'm always thinking about sort of what are better ways to reach kids. We know that neurobiologically, they're wired to think about the present, they're impulsive, they're not thinking about, long-term consequences. Do you have any thoughts about that?
Well, yeah, so I don't think we don't come out with drugs are bad. Right? We do come out with you're at greater risk, and we show basically impact in the brain, right? And so what we're looking for is moderation. We're looking for less binge drinking, and we're telling kids it's worth waiting. The later you start, the less likely you are to get in trouble, right? So it's a little bit of the marshmallow game. Instead of it's 15 minutes, it's like, 25 would be great, wait till you're in graduate school. But, if you start in 6th or 7th or 8th grade, you're really at risk for getting into trouble, right? So kids can often, you're not telling them, you're not showing the fried egg, this is your brain on drugs, because they know that's not true. So you're respecting them, and they're going to try. So you speak to them with that in mind as well.
And I would imagine it would also be important that children have access to things that are pro-social and bring them joy. I mean, I think about, I was just having a conversation with a colleague about how, even just a decade ago at most schools, if you wanted to participate in soccer, football, baseball, that was free and equipment was provided. And now families are needing to pay for soccer cleats and shin guards, and some families just don't have the money. And so those kids.
Not only that, but the activity fee of $300 per sport.
Exactly. Yeah.
Which there are scholarships for, but absolutely. Alternative activities, pro-social activities, physical activity, the arts are very helpful. So they call it, essentially social capital. You've got other things to do and things to lose should you get in trouble. But we successfully denormalized tobacco smoking. tobacco smoking in high school kids is extraordinarily rare at this point. We did great, and now here come vaping. But so real campaigns that really tell the truth to kids, and truth reminds me to talk about basically the Truth Initiative. They were fantastic, and they basically made ads for kids that talked about how they were being used by corporate, corporate greed. They're becoming addicted to something. They were having something marketed to them that was making a lot of money for companies and leaving them with likely a lifelong addiction that would have a 50-50 chance of killing them early. So it was the truth, and it was a very nice angle that showed the kids that they weren't being rebels by smoking. They were really being used by corporations.
Mm-hmm. That's great, and that's a really important fact, because we have seen such an evolution especially in the decreased numbers of people using nicotine. So perhaps some of those tools can be used.
Smoking tobacco, using nicotine via vapes is now going.
Now, yeah, that's a good point.
Right, and then tobacco together with cannabis. Now that cannabis has been sort of normalized, but yeah, so we know we can do it, and I think telling the truth is the main thing. So like having a drink is not going to have a problem, necessarily, but binge drinking can be a major problem. Drinking initiation amongst kids with psychiatric comorbidity and particularly early initiation.
Mm-hmm.
And also cannabis. It looks like starting cannabis in the early teen years may have lasting effects on cognition. Starting cannabis after age 25 does not seem to have lasting negative effects.
Yeah, it's interesting. In my forensic criminal work, I'm often seeing people who have severe addictions, and they started using when they were 6, 7, 8, 9 years old, and it's really interesting to see the trajectory over time. So we're going to pivot. I'm going to ask you some questions about substance use treatment. Okay, so there's a question about psychedelics and whether the future of substance use disorder treatment is psychedelics. What do you think about that?
That's a great question. The jury is out, and it's intriguing. So I'm quite interested in psychedelics for traumatic experiences. We may do a smoking cessation study. We may join in on one. I think we do not know. We so need better treatments, so that I think we're a little bit at risk for believing some of these drugs are magic. I mean, I think cannabis is one where we've sort of thought, maybe it's magic, maybe it does everything as a society. So I'm very interested in that. We don't have to understand the mechanism to see that it works, but we do need well-controlled studies.
And there's some questions, and this is something that I've been wondering as well and hoping for. We have some good medication treatments for opioid use disorder and alcohol use disorder to some extent. Is there anything on the horizon with cocaine use disorder, methamphetamines? I think they've done some limited trials, but is there anything in the pipeline about that sort of extends the availability of medicines to fight these illnesses?
Well, interestingly, not for medicines particularly, nothing worth writing home about, but contingency management looks like it may be getting sort of FDA approval and sort of coverage through Medicaid, Medicare, that's coming. And what that does is shift the decisional balance, right? Maybe those kids, instead of saying you can have a second marshmallow, if those are adults, if you get $30 for a negative test today and $40 in 2 days and $50 in 3 days, that for a relatively inexpensive price, a few $100s, 3 figures, you can get sustained abstinence from some of these disorders. And I think those have, while we continue to look for medication treatments, this is a sort of app-based intervention now that I think holds promise.
That's great, and that could be implemented in a number of different environments. Right. So another question, someone is asking.
Sort of functions like the drug courts, right?
Yes.
A lot to lose, and if you use, and a lot to gain if you don't, right? So it basically helps to shift behavior. so that again, genes aren't destiny, right? You load enough positives on the abstinence side, you really can get abstinence in many people.
So there is actually a question about drug courts, and we may have different opinions on this, but what do you think about, and the question is, what do you think about punishing people for relapse by using jail? What impact is that? And someone asked, what impact is that on the brain? But in general, what are your thoughts just based on the science?
I don't know the science of, I don't know that that's punishing relapse by jail. I do know if there's, I have heard my colleague John Kelly speak about immediate and essentially commensurate consequences to use. So if somebody's wearing an alcohol bracelet and it comes positive, they would spend one night in jail, you know, immediately. So so that would essentially work as contingency management, although it would be a negative reinforcer as opposed to positives, and that does change behavior. I don't know how it would change the brain. I know abstinence changes the brain. So, and with more abstinence, people seem to be able to make less impulsive decisions. So their equipment for decision-making improves with longer abstinence. But what are your thoughts about that?
So it's interesting. The research on drug courts is that for people who are able to successfully get through drug courts, they tend to do better, but the people who are unsuccessful tend to do much worse than they would have done had they not gone to drug court in the first place. And I think just given the impacts of jail on, you know, just family functioning, I do a lot of family work. So when people are, you know, go to jail, even if it's for a night, then that could lead to DCF removing the children from their parents' care, which is a trauma to children and can result in long-term parent-child separation. It may lead them to lose their job if they're not at their job the next day, which can cause other collateral consequences and increase their risk of having long-term substance use disorder. So, you know, I originally was a big fan of drug courts. I think it really depends on the drug court. There's a lot of really good family drug courts. Those don't use any sort of, you know, jail sentences, but they use a lot of incentives and contingency management, and I've seen those be pretty successul.
That sounds fantastic, Stephanie. And, you know, and just to tailor the consequences to be constructive would be super important. So there's got to be no one size fits all. And I certainly know the literature on positive reinforcement as opposed to punishment. And the data on positive reinforcement are great. It doesn't jive with some of our sense as a society, our sense of justice. And that's where I think understanding that you may not be able to empathize as well as you think with somebody who's using drugs, because you might make the right decisions, but they may have different equipment with which they're making their decisions. And I think that's such a crucial point to keep in the front of your mind.
I agree. Okay, we have about 2 more minutes. There's a note that that recovery courts is a better term than drug courts, and I think, you know, I think that sounds great. So there's one question that it's a big question, but I selfishly want to hear the answer because I'm not 100% sure, and I've been thinking about it a lot. But given that substance use disorders sort of negatively change the brain or result in changes of the brain, and the brain, you know, over time starts to heal, and then people will look sort of more neurobiologically similar to you know, control brain, so, you know, someone who's never used than someone with a long-term addiction. Do methadone or Suboxone interfere with that healing, the healing that results from sort of prolonged abstinence?
You know, we've tossed around a study design to really look at that for years. We just haven't found a mechanism to be able to fund it. You know, if we could know ahead of time who was going to successfully succeed, that would be amazing. I wonder if the ABCD trial will, in about 30 years, answer that question. So not going to be able to help you in the next 90 seconds. It's a fantastic question, and we don't know enough. We only have out sort of 3 months for sort of brain data.
So we will wait 30 years. So everyone pause and wait for 30 years. But okay, Eden, this has been such a lovely talk. Thank you so much. We really appreciate your time, and I hope everyone has a really great day.
Thank you so much.